probably get started as the last few folks filter in. Welcome to the Data Science Seminar Series hosted by the eScience Institute. Today we're also jointly the community seminar. I want to circulate these sign-in sheets. This gives us an idea of our breadth and reach across campus and also attendance at our events. There is a column for email address. It's completely optional. You don't have to put your email there. But if you do want to receive more news about eScience activities like this seminar, go ahead and put it in. I'm going to introduce our speaker today. It's Justin Kitzes. He's an assistant professor in biological sciences at the University of Pittsburgh. He received his PhD from the University of California, Berkeley in environmental science, policy, and management. And he was also a Berkeley Institute for Data Science, also known as BIDS Fellow. The Berkeley Institute for Data Science is a partner with eScience on the Moore Sloan Data Science environment, so we have a really close collaboration with that institute. Justin earned his bachelor's degree and master's degrees from Stanford University in Earth Systems, and his research focuses on understanding and predicting species diversity and distributions in human-altered landscapes, as well as applying this knowledge to inform conservation in fragmented habitats. So please join me in welcoming Justin.
All right, well, thank you. So is the microphone on? Does everyone hear it okay? All right, thanks. So thanks, Michaela, for the invitation. I think I know at least some of you from the data science environments, but for those of you who I don't know, my name is Justin, as Michaela said, and I'm going to tell you all about the work that we are doing in our lab, our still relatively new lab at the University of Pittsburgh. So before I get started, actually, how many ecologists or biologists-ish are there in the room right now? That's pretty good. And out of curiosity, for reasons you won't understand until later, are there any cosmologists in the room? Okay. You'll see why I'm asking later. So I probably would best describe my work as quantitative ecology. So I'm going to start off by asking probably the most basic quantitative ecology question of all. How many species are there on Earth? If you're an ecologist, don't answer. How many species are there on Earth? Any guesses? Order of magnitude? Millions. Pretty close. Billions? Probably too high. So eukaryotic species, things with a nucleus, is usually what we try to count. And estimates vary wildly, but I'm going to say it's about 10 million species on Earth. And it's hard, I think, to wrap your head around that amount of diversity. I mean, literally every square inch of the planet almost is covered with a living thing of some kind or another, right? Of various shapes, sizes, evolutionary histories, all sorts of things. Just for a sense of scale for something that's a little easier to grasp, anyone have a guess for how many species of birds there are on Earth? Order of magnitude again? Thousands? Thousands is pretty close. I'll give that to you. Do you know? All right, that's pretty good. Yeah, I'd say about 10,000, order of magnitude, right? 10 or 11,000. Similar for mammals, kind of things we look at a lot, right? Mammals, things with hair, live young, milk. About 5,000 or so species of mammals on the planet. And of course, we have one species with a very outsized influence right now, which is humans. I sometimes remind people that the biological sciences are the discipline charged with the 9.99999 million species that are not the one at the bottom, right? And you probably know, I don't have to tell you, that the other species other than that one at the bottom aren't doing so well right now. So there's increasing evidence that the planet is in the midst of what would be called the 6th mass extinction event in the history of the Earth. The first 5 were caused by either a geological event or an asteroid, and I think we all know what it is that's causing this particular one. And this sort of extinction crisis, if you want to call it that, is sort of the most broad fundamental motivating factor behind the research that we do in the lab. So more specifically, we work on problems surrounding biodiversity loss, which includes the sort of conclusion, the logical conclusion of species extinction, but also population declines, range shifts, and things like that. So that's the main area that we work in. And for practical purposes, I like to divide what we do into sort of 4 major groups of activities or tasks, things you might do surrounding extinction or biodiversity loss. So the first of these is quite simply measurement. How do we know how species populations are changing? How are we actually going to measure that and figure out what's happening out there on the planet? The second is based on that data understanding. Can we use that data to understand what it is that is causing biodiversity loss or population declines? Third is to predict. Can we use that knowledge to predict into the future what might be likely to happen, with the ultimate aim, of course, of fourth, maybe being able to do something actively in terms of management to prevent that from happening. So the different projects that we work on in the lab and that I've worked on over the years basically combine different ones of these tiles in different sort of proportions. But today I'm going to talk about two particular projects that we have going on in the lab. The first one, actually, we're going to start kind of near the bottom here and talk about prediction. So the idea of using, in this case, was basically purely theoretical work, mathematical work, to think about ways of projecting into the future what might happen to species following habitat loss and fragmentation. I'm going to cover this somewhat briefly and then spend more time on the second project, which may be more relevant to eScience, and the data scientists in the room is going to focus more on ways that we go about measuring at large spatiotemporal scales what might be happening to populations on the planet. So that'll take us all the way to the end. So I'm going to jump in first, as I said, with this idea of prediction and talk in a very general theoretical sense about this idea of predicting species extinction following habitat loss or range contraction. I'm going to somewhat hand-wave my way through the math for this, which is probably in all of our interests. But if you have specific questions, I'd be happy to answer them, of course, at the end. So one of the truly canonical patterns in ecology, and one that has a pedigree basically as long as the entire discipline, is the species-area relationship. And it's fairly simple. Imagine I go out into a forest, for example, draw a big square of some area, which we'll call A, count all the species of trees, let's say, inside that plot for some number S, right? Then imagine taking a smaller subset of that area and doing the same thing. You've now got a smaller plot of area A prime. We're going to count all the species in there. That's going to give us some number of species S prime. You can imagine doing this all the way down from small scales up through large scales, and if you put that all together, you get what is the species-area relationship, the relationship between the number of species and the area of the plot that you've sampled. Now, there are lots of different ways that people have tried mathematically for about 100 years to describe this relationship. It's traditionally been viewed as a power law, which I won't get into much here, but what I want to say instead is that recently we've had a lot of success using what is called a sampled species-area relationship construction in order to predict the shape of this curve across particularly very large spatial scales. And I'll just say a few words about what that means to do a sampled species-area relationship or a sampled SAR. It's basically just built on two underlying probability distributions. The first of these call an abundance distribution, the species abundance distribution. Imagine going up to sort of the very largest scale, like the entire Amazon basin, for example, and thinking about the probability that if you picked a random species of tree, that tree would have n individuals at that very large scale, right? So it's the distribution of abundances of species at some very, very large scale. To construct a sampled species-area relationship, we then sort of spatially sample that large-scale abundance distribution with what I would call a plot count distribution. And this is just, you can kind of read it off, the probability that if you had n individuals, so if you had 100 individuals of your species, say, in the entire Amazon basin, that's the probability that in some smaller plot, A prime, you had more than 0, so you were present in that plot, right? And essentially combining those two and summing up gives you a prediction for S prime, the number of species at that smaller scale. And the reason I sort of focus on this is that I think it's been a success story over the years in trying to make predictions about how diversity scales over large areas. And I'll just give you 3 quick examples of that. So this is 3 cases. This is all tree data. So this is the Western Ghats in India, the nation of Panama, and the entire island of Borneo. And in all cases here, we had very good data actually from small plots. So these are sort of 1/4 hectare to 1 hectare plots where someone went out and counted every species of tree found at that very small scale. And the question here, remember on that previous slide I was sort of talking about extinction, you know, a big area gets smaller, you lose some species. We're doing it backwards here. We're looking at a small area and going bigger, but of course it's reversible. If you can go one way, you can go the other way, right? So in this case, we had this very small scale data, and we also had species lists for the entire island, nation, or region, right? And so what we wanted to do is see whether we could use just the data from those very small plots and upscale to those other ones. And this sampled SAR construction actually does very well. Now, of course, I say very well. You say, Justin, come on, you made the scale all funny, so it looks like you got really close, right? But just for a sense of comparison, so that traditional species-area relationship, as I mentioned, the power law would have given a result that was something like this. So this is much, much, much closer than what you would be able to do with what is traditionally assumed to be the form of this relationship. So as I said, for me, this has actually been something of a success story. We've actually done a pretty good job of thinking about going up. And as I mentioned, you can also think about this as going down. So for example, if you were to say, you were going to lose 3/4 of the forest habitat in Panama, you would take this number, read it sort of 3/4 the way down, and say this is how many species you would have remaining after that habitat loss, right? So that would be how you would read it for extinction. So as I said, this is working very well, but there is sort of a kind of a trick or an assumption or a missing piece here, which is that, you remember in that previous slide I showed that big square going down to that little square, right? That's not actually how habitat is lost in practice. This is more what things actually look like. So this is the island of Borneo. The light green there is forest that was logged, so no longer intact forest, and the dark green is the intact area. And you can see in this case it sort of shrank down to something that was more or less one large area-ish, but that's obviously not a square. In fact, if you look more carefully, you can see that there's quite a few sort of isolated pieces of habitat that are off by themselves in these various little areas. And this is pretty common. I mean, this is the way that habitat loss actually occurs in practice. And so what would be much more useful than having something which was really predicated on the idea of one big area shrinking to one smaller area would be to try to have or derive a relationship that would talk about multiple patches. So rather than thinking about one large area of habitat shrinking down, we could think about fragments and changes in the sizes of those individual patches, maybe getting smaller, maybe blinking out of existence entirely, or in the event that you're a land trust or an open space district or something like that, thinking about adding patches to a network to try to increase the number of species that were protected in a network of reserves or something like that, right? So this is one of the very first problems I started working on when I started my PhD. So I've been thinking about this on and off for almost a decade, basically. And to give you a sense, we tried half a dozen different ways of doing this to try to basically construct the one on the right from the same assumptions as the one on the left. And to give you some idea of how unclear we were about this, at various times during those 10 years, I spent time trying to show how to do it, and then spent time trying to prove that it was impossible. And eventually we did figure out that it was possible. And the reason it was so hard, at least for me to think about this, is that you have to take a detour through a relationship that you wouldn't initially think was relevant here. And you'll see why I asked if there were any cosmologists here. So the beauty of this, at least from your perspective, is I get to condense all that work into 2 slides. You don't have to sit with me for the whole time and actually work it out. So here's eventually what we came out with. So I'm going to start by simplifying the species area problem a little bit, and just focusing on that second distribution we saw on the previous slide, the plot count distribution, just the one that's the spatial part, right? And we're only going to think about a single species. Now, we can think about constructing that plot count distribution by taking, say, a large kind of landscape like that and gridding it up into smaller squares, and then looking at the distribution of counts of individuals across those squares. That's one way of just measuring the shape of this plot count distribution, right? The probability that you pick a plot and it's got some number n individuals in it. So now let's think about something else called the pair correlation function. So to construct this function, this is well known to cosmologists, we pick out, basically imagine all those dots are individuals. Pick out one dot and stand on top of it. And then imagine walking out some distance d and drawing a circle. The correlation function is proportional, technically, to the probability that you get another individual at some distance d away, right? So you can kind of think of it like the probability of getting a double hit between the individual that you're standing on, and then you get another tree of that species at some distance away, right? The correlation function tends to be peaked and monotonically decreasing for the most part. Individuals of a species tend to be aggregated, kind of clumped together in space. And what that means is that at short distances, you have a higher probability of getting that double hit, right? And as you get farther and farther away, it eventually decays down to a baseline where it's uncorrelated, the presence of the 2 species as you get, 2 individuals, excuse me, as you get farther and farther away. So the whole trick to this multi-patch species area relationship turns out to be that you can relate these 2 functions to each other. So the long and the short of it is, let's start with the plot count distribution. Notice that we can calculate from just a square plot like that a seemingly unusual number to start with, which is the expected number of pairs of individuals. And that would be just the average of n times n minus 1, right? You imagine taking every individual, going to every other one. Take another individual, go to every other one, right? And that's the expected number of pairs in the plot, which it turns out you can express as a function of the mean and the variance of that plot count distribution. So the important thing, really the nugget that took so long to figure out, was this. You can also imagine going to that same plot and gridding it up into very, very, very small squares, so small that you can only get one individual or 0 inside of one of those little tiny dots. Take a little red box and move it all around. And for each place you put that red dot, take a little orange, maybe it's orange box, and move it all around also. Every time you've got two of those tiny boxes, you've got some distance d that they're separated by. The integral of the correlation function over all possible locations of those two little boxes gives you the same thing, the expected number of pairs of individuals in that plot. Remember, the correlation function was that probability of a double hit. So take every possible location of those two little boxes, count up the double hits, and that's also the number of pairs. In practice, this is a quadruple integral over the two xy coordinates of the two boxes, right? For those of you who don't like the quadruple integral, I promise this is the last slide with any math on it. So I just want to show then how you sort of hopscotch from that one kind of key point all the way down to this multi-patch species area relationship. So the first thing is what I just showed. Use that expected number of pairs to relate the mean and the variance of the spatial plot count distribution to that integral of the correlation function. It turns out if you take now two patches that are separated from each other and put that little red box over in this patch only and put the little orange box in this patch, you can do that integral again, and you end up with the covariance in abundance between those two. You can take that covariance between every possible pair of patches in a whole network, gives you a covariance matrix. The means are actually quite simple to come by, and you can get a multi-patch version of that point count distribution, which then you can plug back in to the same equation we saw before and actually get a multi-patch species area relationship. What I find so interesting about this, and this was what we spent so long trying to think through, is that there was actually enough information, almost, with a little assumption or two, there was almost enough information embedded already in the univariate species area relationship, the regular one, to actually do the multi-patch version. You just had to be kind of clever about how to pull it out. But the same set of assumptions actually give you both of them for the most part. So that's essentially, I mean, that entire story there is we did eventually find that you could do this, and we're still in the process of working this up right now. And as I mentioned, I think this multi-patch species area relationship is a lot more relevant to this kind of practical case that we have down here where habitat's lost in some sort of fragmented way. So that's all I wanted to say about sort of some of the major theoretical work that we're doing in the lab right now. And now I want to turn over to, again, things that may be a little more interesting for the data scientists in the room and talk about our efforts to come back to the beginning of this set of tiles here and talk really about measurement of species richness, of population change over time. And the way that we do this in our lab is we spend a lot of time on acoustic analysis. That's a picture of an acoustic recorder right there, which I'll talk about quite a bit. But before jumping into the specifics of the acoustic work that we do, I want to talk a little bit about why it is that we even need new ways of measuring biodiversity on the planet. There's no question that we're living in an age where we have more data available on biodiversity than we ever have before. And one often cited example of this is the Global Biodiversity Information Facility, the GBIF database, which collates point records from a number of different databases collected by people all around the world. So because we're going to talk a lot about point occurrence records, I just want to be very clear. This is basically a geospatial data point. So I saw an individual of a species right here at this time. That's what one of these point records is, right? And GBIF, mostly through the efforts of citizen science projects like eBird, has managed to collect about 1 billion of these records scattered all around the world over about the past 100 years or so. And whenever I say this, I often get like, wow, that's a lot of data. Well, I want to push a little bit on that feeling, that this feels like a lot of information. So if you sort of break this down a little bit, I'm just going to focus on the US here, although everything extrapolates the same, if not even slightly worse when you talk about the globe. GBIF is currently adding about 50 million data points every year in the continental United States. Now let's just back of the envelope. Assume very generously that each one of those observations covers about 100 square meters. Might be a little bit of an underestimate for a bird. It's definitely big for a plant, right? And let's say that by virtue of the observation, someone actually being present in the field, they can guarantee that that species was at that point maybe for about a 10-minute window or so, right? If you actually just multiply that out, what you find is 5,000 square kilometers. That's how big 5,000 square kilometers is on a map of the United States. So what I'm saying is that despite all of these efforts and what feels like a lot of data, the effective area outside of that red dot is effectively unobserved for wildlife in the United States. Now, the exception potentially being satellite imagery and the possibility of doing maybe tree species, large vegetation analysis from that, but certainly for things that are too small to see from space, this remains true and I think is going to continue to remain true with the approaches that we have today. I'll just point out that if you include the time dimension, of course, everything looks even worse. If you include the species dimension, it looks even worse. Now, that's not to say that this data is useless. I don't mean to imply that at all. There are certainly ways of extrapolating or scaling up information like this to large spatial scales. That works particularly well if the data is randomly sampled and unbiased, which it is not. But even so, there are still a lot of questions you can answer by making extrapolation. What I want to talk more specifically about, though, are the types of questions or problems where you just have to go out and see what's happening, right? You can build models, you can make extrapolation, but at some point at the end of the day, you just got to go out there and look at what is occurring. And that's the challenge that we are having a very hard time with right now, is that just basic act of observation. So can we just go out and get more data? Well, on the one hand, no. Because you could take every ecologist in the United States, have them do nothing but sample in the field for their entire working life, and you would get up to like 0.3% or something like that. So you would triple the amount of the area coverage up to 0.3%. This already includes all of those citizen science efforts. So you triple, quadruple the citizen science efforts, you're still well under 1% of the area of the continental United States. And again, it's not to say this isn't useful in some ways, but the basic point is that direct biodiversity observation does not scale. There aren't enough eyeballs and boots to actually accomplish what we might want to accomplish, right? It's probably no surprise to many of you in the room that there is another way we could approach this, which is what I would call indirect observation. So this is technologically mediated observation where you are no longer rate limited by the number of human eyeballs that you can send out into the field. And there's many different ways of doing this. That's an acoustic recorder, which I'll talk quite a bit about. This is a camera trap in the middle. There's actually a drone that was designed to deliver mosquito traps. And you know, this shift to indirect observation has a lot of precedence in other areas of science. So if you think about what happened in astronomy and cosmology when you had automated sky surveys, or what happened to geography and planning when you started having satellite imagery available, this is the same kind of shift that I feel very sure ecology is currently in the midst of going through. So just because I can, because I'm standing up here, I'll sort of make my little speculation line in the sand. I would wager that whether you like it or not, which is an important point because there are pros and cons to this, whether you like it or not, in 40 years, which I picked sort of, you know, just to be, that's how long it takes the entire population of faculty to turn over in a university, basically. So in one academic generation, approximately, I'm going to wager that over 99% of the data we have on biodiversity is going to be collected by these means indirectly with automated processing, and then maybe in some cases reviewed by humans. This is a big, big, big difference from the way that we're doing business right now, and we're just starting to see this start to occur. And I want to talk a little bit about the part of this that our lab works on. But before I do that, actually, I want to say a word about some of our previous research projects very briefly to illustrate sort of what the state of the art was in this area just maybe 5 years ago, and as a contrast to the kinds of things that we're working on now. So to talk about that previous work, we are going to talk about bats, because bats are very interesting for any number of reasons. They're not at all scary, I promise. I've held a bat, it's fine, didn't attack me. But there's two particularly salient points regarding bats when it comes to indirect survey, and particularly acoustics. The first is that bats are really a pain to study by traditional means. You got to go out at night, you set up a big mist net, you wait and you wait and you wait. The bat comes, he flies around the mist net because they can see the net, and he keeps going. But they're also, conversely, a very good candidate for acoustic surveys, because many of you may know that bats echolocate, right? So the way that they feed is they fly around sending out what are essentially little sonar pulses and listening for the reflections off of their prey, which is how they forage on the wing. So it's a very good system for us to eavesdrop on to hear whether a bat is present or not. The other particularly useful thing about bats, as opposed to other things that make noise that we can record, is that these calls are functional. So they're tuned to the type and the size of the prey that they're seeking, and also related to their evolved body size and sort of physical structure. So what that means, actually, is that the calls are very consistent within a species, and in many cases quite different across species, which makes for a relatively easy system to use to distinguish species from each other from this indirect type of data. So again, I just want to say very briefly, give you a sense for the type of work that we did on this. We conducted 2 studies when I was back in California before I moved to Pittsburgh. Both of these were designed to look at the effects of land use, human land use, on bat populations, bat activity, I should say, and across different types of species. First of these studies was up here in Napa and Sonoma County. We were studying the effects of agricultural land conversion on bat activity. So we did edge interior pairs at vineyards up there. This was not, I will admit, a bad place to do field work. My wife would come with me. We'd hang some detectors, go to a winery, hang some more detectors, come home. It really wasn't that bad. The second study was on road effects. So these were 3 large highways crossing the San Francisco Bay. And we were running transects perpendicular to the roads, so looking at whether there was foraging activity near the roads versus at a control point farther from the roads. In contrast, this was one of the worst possible places to do field work. If you've ever stood by the side of a freeway in Oakland, California at night, you might have some idea why that's a bad idea. To be fair, Oakland is only the 6th most dangerous city according to the FBI, but still, not as good as the first one. If there is anybody in this room who somehow has not heard of this book yet, I wanted to bring it to your attention. So in addition to the publications on this, this was a book that I co-edited with some other colleagues at Berkeley when I was at Berkeley. There is a case study in here, one of my contributed chapters, that actually talks about this workflow in a little more detail than I'll give today. You can actually find the whole book online. It's mostly focused on reproducible research and open science. But for the purposes of today, we don't really need to talk about the whole workflow. I'm just going to divide this kind of into the 2 major sort of really data science parts of the pipeline, which is dealing with the actual act of audio recording in the field, which is essentially a hardware problem, and then the act of doing species identification from that data that you bring back, which at heart is some type of machine learning problem. So to start off, for the actual recording, basically what you need is a microphone, a sound card, and a battery that you can put out in the field, right? We need an ultrasonic sound card, high-frequency sound card, of course. But that's what it boils down to. These were the 2 types of hardware setups that we used for this study. The one on the left is a kind of a custom Anabat system, and the one on the right is a song meter. The wildlife acoustic song meter on the right is, I would say, almost for sure the most common field recorder still in use today. Costs, you can see, about $2,500 to $1,000. At that price point, we had about 8 of these detectors at a time that we would sort of rotate from site to site in order to take this data. You had to charge the batteries maybe once a week or so, depending on how long you were recording. So this is how we bring the data back. Now, once you bring the data back, what are you going to do with that? You remember the goal is to generate something that looks like a point occurrence record, right? We want to see what bat was where at what time, basically, and then we'd maybe sum them up over the night or something like that. But that type of occurrence is the basic goal. The way we almost always do this, actually, regardless of the species, birds, bats, frogs, marine organisms, anything like that, is to convert things into spectrograms, and then it becomes sort of an image classification problem, actually. So in the case of bats, there's very little noise in ultrasonic frequencies, and we tend to get very clear patterns like this, these nice little kind of like hockey sticks, which are fairly easy to pull out. Each of these, again, is a sonar pulse that's sent out by the bat as it's foraging for its prey. So what we did here is we extracted, this was using a free piece of software that was closed source, however, about 11 parametric features, parametric in quotes, of these calls. So these are things that are fairly straightforward. These are pretty clean signals. Maximum frequency, minimum frequency, duration, slope, location of the knee. It's kind of like a little inflection point over there, things like that. And then we just fed that into a random forest. Of course, you have labeled data you use to train the model this way, and then you take your unlabeled data and use it to predict on that. And you get back out things that we want, such as the number of calls per species per night at some sampling point, right? And like I said, these calls are pretty clean. Depends a little bit on where you are and things like that. But just doing something naive like this in Python, we got almost 90% accuracy, doing nothing else, no other tuning, nothing else fancy, right? So bat calls are a relatively easy, as these things go, classification problem. So when we did all that, we used GLMMs, kind of all the usual stuff. What do we find? So the agricultural effects, for example, as expected, you get a lot more bat calls at the edges of these vineyards. The effect was weaker for higher foraging species, which makes some sense. Probably the most interesting thing we found was that for a lot of species, you have landscape effects. So it's not just what's at the point that you're standing on, but what the habitat looks like in larger surrounding areas around that influence what you see. For bats, we didn't really see that landscape effect, which is unusual. It's probably related to their foraging behavior. Road effects, only about half as many bats near roads. All species were affected pretty equally. That road effect was actually temperature dependent, which was a somewhat interesting finding. On warm nights, basically, they didn't avoid the road as much as they did on cold nights. I don't, unfortunately, have a good explanation for that, but it's worth more follow-up. So that's sort of, again, to give you some sense of where we started with this, but also kind of what the state of the art was not that long ago, right? So going forward, the type of work that we're doing in our lab, so what we really want to do is go beyond the scale of a single study, single site, few detectors, kind of the way that we would normally do this type of work. So again, when I'm have grad students show up or something like that, I tell them something that sounds like this. This is what we want to do, right? Next year, we want to have 1,000 recording units out in the field simultaneously, recording bird and bat calls, 3+ hours per day, depending on the application, all the species automatically ID'd using open source classification software, right? So you may wonder, how in the world are we going to get from what I just showed you to what's written on this slide, right? So I'm going to tell you how in the world we're going to do that, at least how we're in the process of getting there. So the first thing that really breaks this open is the hardware. So over the years, as you saw, that hardware, the song meters are $1,000, $800. You might wonder to yourself, why can I buy a Raspberry Pi for $5 and it costs $1,000 to buy something that has a microphone attached to it? I would say you are correct. That is a very good question. And so we played around over the years with Android phones, with Raspberry Pis and things like that, but power was always kind of an issue. And what really finally broke this open was last year a group at the University of Southampton in the UK released an open source schematic for something that they call the audio Moth. And we have about 400 of these in the lab right now because you can print them for about $50. So you can't buy these commercially, but it's just, I actually have one over here if anyone wants to see it after. It's about yay big, and it's just a little circuit board. The mic is soldered right to the board. It's got a battery pack attached to the back, SD card that saves the data. And as I said, you can print these for about $50 now. So, like I said, we have 400 of them. You think about that, putting out 1,000 sounds like a lot. At $50 a pop, that's $50,000. That's well within a single PI grant, right, to put out 1,000 of these units. Yeah, do you have a question?
That's not the main part of the cost. You got to go in the forest and hang on.
You'd be surprised how cheap undergrads are, but we could talk about that after. None of these are networked. No, none of these are networked. We could talk about that after you'd like. So but the important thing is that once you get to that scale, you're recording a lot of data. So depending on the relative sort of proportions of bird and bat recording that we're talking easily hundreds of terabytes, possibly up to a petabyte if we're doing a lot of bat recording over the course of a year. And that generates, if you add it up, on the order of, I'm estimating here, sort of 100 million or more point occurrence records. Remember how many point occurrence records are going into GBIF every year in the United States? 50 million. This one single lab deployment is going to double, triple, quadruple that number. And this isn't to toot our own horn, although I wouldn't mind, but this isn't to toot our own horn. This is to say that this is well within the scope of what a single lab can generate at this point. Multiple times the total number of observations that we are getting this year in the GBIF database. Again, this is a radical change in the scale of the data that we're looking at for these types of questions. So that's the hardware problem. The software problem, of course, is still there. When you got all this data, how are you going to actually do species identification from this? So this is something we're working on. We have financial support from a couple sources to work on a piece of software we call Open Soundscape. We eventually want this to be a platform for different types of acoustic event classifiers to be built in. We're starting with something I'll show you in a second. But the idea here is that not only is this open source software, but it's scalable. So we basically are designing it to be put in containers with the idea that you can run it on your laptop for small applications, you can run it on a cluster, you can run it on a supercomputer if you have to, right? Depending on the scale of the data that you're working with, commercial cloud service, et cetera, et cetera. So that's the sort of platform that we're using. Now, I switched kind of subtly without really telling you from bats to birds, right? Birds are a very interesting problem because unlike bat calls, which are very clean, if you remember I showed you this, bat calls are very clean. You can sort of easily say, oh, min frequency, max frequency, and that gives you a pretty good description of the shape of that hockey stick. All these calls are basically hockey sticks, right? Birds are much more complicated. I don't know if this is going to play. I brought one with me, so I'm talking about birds. There you go. So that's just a common yellowthroat. And you can sort of see in the spectrogram representation here, that's a much more complicated shape than a hockey stick, right? And so the first challenge you kind of have to deal with is how are you going to come up with a low-dimensional representation of the sound in order to try to distinguish, say, this species from other species that would co-occur with it, right? And oops, let's start playing again. So fortunately for us, in terms of algorithms, there has been a lot of work on this over the years. And so we're sort of following the literature at this point on bird call classification. So the very first of these competitions that I'm aware of was a NIPS competition in 2013. It was followed up by something called Life Clef, or Bird Clef. This is actually very interesting. If you're interested in this stuff, I would encourage you to Google this challenge. They have a number of different, basically, image recognition type challenges. But the thing I want to point out is, you see down here, who's the winner? Mario, right? There's this guy named Mario Lasek. I don't know him. I've never talked to him. He's at a museum in Berlin. He's won every single one of these competitions, except the year that he took a break, and I'll come back to that in a little bit. He's won every single one of these competitions, and he's basically won it with the same method every year, regardless of which data set they've used, how big it's been, et cetera, et cetera. So a very logical place to start if one is trying to do this. So I'll just say a word about this kind of clever idea. It's also worth pointing out that I believe this was actually pioneered for whales. So it actually started in the marine realm, and he borrowed it from there before applying it to the case of birds. So this is sort of his answer to this low-dimensional representation, which is based on template matching using cross-correlations. It's actually fairly straightforward, if very clever. So this is, say, for example, our spectrogram over here. For example purposes, that was, again, a common yellowthroat. We start by taking templates over here. These are just syllables extracted from a whole variety of birdsong files from different species. All you do is take those templates and slide them across this unknown file, looking for the maximum cross-correlation that you get at any point. Remember, the x dimension is time, so it doesn't really matter where you were in time. Y dimension is frequency, so you don't want to move the y dimension. And when you do that, you get a cross-correlation vector, which is just how well each of those templates matched some point in this file. So, of course, if it's a very close match, you get a very high cross-correlation. This doesn't really appear anywhere in there, so it's very low, maybe something intermediate. The whole idea is basically that that cross-correlation vector becomes your feature vector, which you then throw back into another random forest, just like we talked about with the bats, and end up generating the same thing you would before. So like I said, it's sort of a clever way of getting around that problem of how to represent those calls in some low-dimensional space. You're just representing them by how well they match these little templates that you've picked out. So one final word on this is that I mentioned before that Mario Lasek won every one of these competitions except for the year he took a break. So he used that method up until 2017. He took a break. And I remember, I'm looking at the competition. I was like, it's interesting. He took a break. I wonder where he went. Well, the answer was he came back the following year with a CNN and won again. So the field is finally at the point where we're starting to shift towards deep CNNs instead of things like template matching. It's worth pointing out that template matching beat neural nets for a long time. But the thing actually that I think is very interesting about this, so this is using, I think it was the Inception model. So this is on top of models that were pre-trained on the ImageNet data, right? So this is an image classification problem, essentially. The interesting thing about this is that the other thing that helped make this viable, I think, finally, is data augmentation. So what folks have finally started doing is taking what is actually a very limited set of training data, of labeled birdsong, let's say, and augmenting it by degrading it, stretching it, squishing it, rotating a little bit, and creating synthetic soundscapes that contain all sorts of different species and all sorts of different combinations under different simulated conditions, and training on that, which sort of gives you enough breadth to cover what you're actually going to see in the field. And for my money, I think that's probably what is driving a lot of this improvement, not the neural net itself, really. So finally, I want to say one last thing about this, which is, even with all of these improvements, I mentioned before that for back calls, a naive method got 90% accuracy already, right? Birds are much, much harder. If you have a nice, clean signal, like the one I showed before, a bird classifier in this competition will win with something like 70% accuracy or 80% accuracy, which isn't bad. When you start to get calls overlapping in complicated soundscapes with noise and other things like that, the winning entries in these competitions are winning with accuracies around 20%. So that's a much, much, much harder problem when you've got all these things on top of each other. And I'll say in a little bit some things we're thinking about to improve that. But in the meantime, I think one thing, I don't expect you to read this, but I think one thing that's very important is to start thinking about treating uncertainty in these call classifiers very explicitly. And this is something I think we're not doing a very good job of in ecology right now. So I put this up only to say that this is actually not that different from a type of statistical model you would normally run to figure something out about birds in the field. You have essentially the true presence or absence, which is related to some environmental characteristics. So you got environmental characteristics to tell you whether a yellowthroat is there. You've then got a layer that has to do with observation and sort of how well you can detect whether the yellowthroat is there. That's all pretty normal stuff. But the key that I want to point out is this bottom layer. And for some reason, I don't see people doing this. All we did here was take the probability of the presence of a yellowthroat as the input data rather than a binary 1 or 0. So usually what people do with these models is you'll threshold whatever comes out so you get presence or absence, and then you'll go forward from there. And the only point I want to make here is there's really no good reason we shouldn't be taking the probability that a species is present or absent in a given place as the data that we work with. So for example, when you decide to say that yellowthroats prefer riparian areas, you're doing that in light of the fact that you never exactly knew where the yellowthroats were in all of these recordings. There was always some uncertainty in that initial classification. So I just want to give a sense of some of the places we're putting these recorders out in the field right now. So as I said, 1,000 units, we're going to be expanding, but this is some of the work we've started to do so far. North Central PA, we were on a road transect here. You asked about putting these things out. If you're along a road, it's a lot faster. So we had 200 recorders put out along a 20-kilometer transect along a state forest road to look at sort of small scale to landscape scale scaling patterns in bird communities. This was working great until people started shooting them. I can talk about that later if you're interested. We also took out some recorders to Flight 93 National Memorial. I have a hopefully soon to be starting graduate student with me who's interested in surface mine reclamation. And so we were looking at the use of use of these reclaimed ponds versus these natural riparian areas. It turns out they're actually using this stuff, which I think was sort of surprising to everybody. We've had these up on a pole in southwestern Pennsylvania along with several other recorders listening to nocturnal flight calls. This is a very common way of tracking bird migration, is to listen to the calls they make as they fly overhead at night. So we have some up there. We just had a collaborator take about 20 of these to Borneo, put them on the side of different streams, so some that were in intact forest, some that were in fragmented or log forest, to listen to the amphibian communities there. So this is some of the first places that we're starting to do some of this work. And I just want to close by pointing a little bit to one last sort of project that we're just starting now, which is focused on a problem that we have with these recorders. So when you stick up a single microphone like that, you are listening to everything in maybe a 3-hectare circle, right? It's all on top of each other. It's all sort of complicated. But the most important thing is that you can't distinguish individual birds when you do that. If you want this type of technology to replace a human in the field, when humans go out, they will take a density estimate. They will tell you how many yellowthroats were out there because they can hear them in different places, right? And that's something we can't do with this design. So we're starting to try to get around that. And the way to do it is with time-synchronized arrays of recorders. So on the hardware side, we have some folks in the electronics shop who are working with us to attach GPS units to these audio moths. That's actually a Raspberry Pi we're using as a time server. The Moth is back there at this point. But the idea here isn't to get position from the GPS, it's to sync the clocks. Because if you sync the clocks, you can actually use the fact that sound does not travel instantaneously to localize where the sound came from. So this is, for example, a 4-track recording. This is an Eastern Towhee. And you're basically using that little bit of difference in the time of arrival of the sound between these different recorders along with their positions to figure out where the origin of that sound was. This came from my tech. I know it's simulated data because that's too clean right there. So every pair of recorders you get defines a hyperbola upon which that call had to be located in order to give exactly that difference in time between where it arrived at the two. If you have 2 pairs, you get 2 hyperbola, and you get an intersection. If you have multiple, of course, what you're looking for is the intersection of that point. In real data, it's not one point. You get a little cloud, and you sort of take the average, right? But that's the basic idea. This is something that we're just starting to work on now, and I think is really one of the key points to enabling this type of work to actually replace humans for some of these surveys in the field. So that's essentially all that I have. I want to thank many of our collaborators, particularly members of my lab there in the top left, Joe Begley in the electronics shop, Barry Moore at the Center for Research Computing at Pitt. We're collaborating with many organizations on the top there on the field work, and we've been supported from various financial sources over the years. I'd be happy to take any questions. Thank you. Yeah.
So that was great. I could listen to your talk. I know of another large source of auditory data, which has been just countered a lot of these problems as you described it, as the National Park Service has been putting out like reporters of all the national parks for years and years. And a friend of mine is working on that data and says we just don't have labels. Like we just don't know what things sound like, and so we can either employ 30 undergrads for 2 summers to listen to audio until their ears fall off, or do something smarter. So I'm wondering what the state of the art is on some sort of clustering or identifying clusters of similar sounds to generate data.
Yeah.
We don't have thousands of bird calls in a database.
Yeah, in the event that you need to generate label data, certainly unsupervised learning, so clustering type algorithms work here. You know, probably, and I believe the Park Service has already done this. I mean, so the easiest, if one has this kind of data, the easiest thing to do is to look at soundscape indices. So these are just indices of like relative activity, amplitude, coverage of frequency bands, things like that. And a lot of folks do that to look at sort of just the relative soundscape patterns between different locations. That's probably the easiest. You know, I actually think that with some of the data augmentation, the labeling is not as big of an issue. My intuition is that if you took something like the template matching approach, which is fairly kind of non-parametric, you don't have to think a lot about that, it's just cross correlations, and combine that with data augmentation, you could actually start with a pretty small labeling data set and get pretty far. I mean, maybe not to 100% accuracy, but that's where I think would probably be the first place to look in that respect, before employing 30 people for 10 years or whatever. Yeah. Yes?
That abstract that you showed from the paper from
Mm-hmm.
Mario, it seemed to indicate that he was looking at 1,500 species.
Yeah.
And so I'm wondering if the classification is better, because you said it was like 20%.
Yeah.
Is it better if you say, hey, I know I only have 100 species here, and you do better, or is it just that bird calls are not?
I think you would do better. I can't answer that question because I haven't tried, but once we try, I will let you know. I think it will get a lot better. I mean, really the problem is related to the number of species, but it's really the number of sort of mimics or similar species. So if you can rule out species that are similar to each other, then yes, I think you would perform quite a bit better. Yep.
I had a question about, for example, if you place one of your microphones in a rainforest, for example, and there's a lot of different sounds, and maybe some of the species are within the same frequency. How do you isolate which?
It's very hard to do. And actually, I mean, with a single microphone, you really can't do that because everything comes in squashed on top of each other. So that's the kind of case where having an array like this is actually very important, because what you could do in principle in a case like this is you take every little syllable, every little sound that you get, and you map it to a point in space. And so in theory, the idea would be you could say, like, look down at the map and draw a little polygon and say, just give me everything that was making noise in that square, right? And that reduces down sort of the overlap issue from 3 hectares to, 10 square meters, 50 square meters, something like that, right? So I think ultimately that's probably the solution to a problem like that. The source separation needs to be done with an array. Yeah?
Do these have to be tuned for a certain frequency range, or can you get like a wide swath?
They're recording everything, ambient sound, up to the frequency determined by the frequency of your sampling. Yeah.
How much maintenance effort did you put into recorders? Like how much effort you need to maintain them?
Yeah. Not very much. I mean, a lot of it depends on how much loss you're willing to accept. I mean, at $50, we could lose some. It's okay. I mean, I think this was the only picture I have in actual deployment, but that's a Ziploc bag duct taped to a tree. So not particularly heavy in terms of, intellectual effort or cost in terms of housing them. And they will actually survive at least a month or two. More than degradation is whether you have any interested animals. That's more the problem. If a bear wants your box, the bear's got your box. It doesn't matter what you do, but again, I think, we've had equipment loss with really expensive weatherproofed ones also, I think more to the point, this is sort of the mentality where you've got high turnover, cheap, high throughput. You're going to lose 10% of them, fine, put 10% more out, type of thing. Yeah.
So you mentioned they were being shot at?
Yeah. So apparently it's a good story. So we were called by the forester 3 or 4 days after. Not me. We were called by the district forester at that forest 3 or 4 days after we put those out, and he said, "Oh, you guys." He said, so apparently, like, the rumor at the local bar was that we were the game commission doing a road hunting sting. So we had a couple of them shot, which, okay, whatever. But we also had a couple of them which were very nicely taken off the tree with the batteries out and placed very gently on the ground. So, I don't want to mess with your stuff, just don't record me, So that's just hazards of working on public lands, basically. Yeah. I would love to. In the lab, we called this the Blue Apron model, where you send people a box of 20 of them, tell them to put them out, and just mail them back to me. We'll mail you a new one, I think it would be a great idea. We're not at that point yet, basically. Yeah. Yeah.
You're getting a lot of sound. I mean, if you're
In terms of regulatory authorities, that's a good question. I actually don't know what the legal ramifications are exactly in that respect. But in terms of, I mean, there are people who do chainsaw detection and gunshot detection specifically. Usually for those applications, they're spacing them a lot farther apart, and they are cellular linked or something like that, because if you hear a chainsaw, you want real-time information on that so you can go there, right? You know, we're bringing back this data on a 1 to 2 month horizon for the most part. When we're doing birds, it's probably too slow for most of that. Not that you couldn't get indices of human activity. We could certainly do that.